Okay, so I'm going to switch gears here, and I've got two data sets that I would like to show. Um, the first one being the RNA Seq data set, and as part of it, it has a Genome Browser baked into it. And then the second one is an imaging data set, and that one has the OrthoSlice viewer. So let me just switch over to that. Um, okay, and let's see, are we seeing my browser here? Okay,
Yes.
Chris is nodding yes. So okay. So now here is an RNA seq, a data set. And again, what we have at the top here, I'm just going to be a bit repetitive because that just helps with catching the information here. But again, we have at the top here, we have our identifier section and our preferred identifier, the digital object identifier, DOI here. We see also when this was released, so we have an idea of about how long ago this was produced. We also have a brief description of the data set and then a link back to the project. And so, like I said, if we were to find our way here, we might then just want to go to this project page. I'll open this in another tab and just see what else they've produced. And so if I go to that project page on our site, we get an abstract, so we get an idea of what their overall inquiry was about. And then beneath that, we see the 18 in total, 17 other data sets that have been produced. And it's likely that there is related and relevant information if we were interested in the first data set we were looking at. So let me just go back to that page. And then, as I mentioned, what follows after the attribution detail, so we have the contributors right here, are the categories of keywords. And there's a little button over here on the right-hand side called Table Notes. I switched that, and what I can see are the details of the terminology. So I saw it was an RNA Seq assay. We're probably all very clear with what that means, but if there's any uncertainty about a term, we can look at how it was described. So we have the description, and also under the ID, you'll see this is not a FaceBase accession number. It says OBI and then a number, and this comes from the Ontology of Biomedical Investigations. So we try to source these terms from outside standards as much as possible. So I can kind of go down here and see that the species comes from the NCBI taxon. And let's see, anatomy is from the Uberon ontology, which is kind of an overall cross-referencing ontology from other sources. So that kind of information is available here. And again, when there's not a suitable external term, we pull from or we'll have internally or FaceBase defined terms as needed. And we move down here, we've got our Genome Browser page. So this window is embedded here. If you're familiar with the Genome Browser, you know that you can navigate to other chromosomal locations. This is just coming up at a default location. Let's see, and I'll come back to this and open it up in another window. But if we scroll down, this is a rather large data set, so we have quite a bit in there. But we also have FaceBase Hub, and we have the other super tracks that are available from FaceBase for this particular assembly. So this is an MM10 assembly that this data is based on. So there's other data for MM9 for the human assemblies, but this is the collection. So we can turn these on and show them as well as other standard data within the Genome Browser. Then next we get down to the experiment and biosample. And at this point, let me just point out on the left-hand side this sections listing here. So this is kind of a table of contents for the data set. And what we have are beneath the summary, we have the Genome Browser, and then we see that we have experiment. There are going to be 8 records there. Biosample, sequencing data, process data, track data. So that's what we can expect in this data set as we go down here. So in experiment, we see that we have, we're displaying all 8 records. So we have 8 different experimental records here. These correspond to like one specific RNA-seq assay, performed over a group of replicates. So the first 4 here we can see were the controls, and the next 4 were the mutant groups. And then as we move down to biosamples, we see that we're looking at the first 25, okay, of biosamples. So we look at these, we can kind of get an idea of additional biosample related information. And we see that there's a little extra detail beyond the anatomy. So we have intermaxillary suture, and we have a maxillary osteogenic front, and also suture mesenchyme data. So we have this for well, I don't know the number yet, but we'll see the number. But for this collection of samples, and we can see that we have some different age stages. Now, if I want to see more right here on the page, I can use this drop down, and I'll just bump it up to 50. And now it loads, and it says that it's displaying all 38. So if I want to see everything right away, I can get an idea of all that is in this data set immediately. Now, you can also drill down further and just explore this data a bit. You know, let's say I wanted to filter and just wanted to see one specific age stage, what was in there. I can go to this explore link, and now we're actually back in the search display that Chris was initially showing us. And here, but I'm just searching within this data set. So we'll see up at the top here, I have one little indicator of a current filter that's been set, and it's set on that data set. So I'm just looking within this one data set, but on the left-hand side, I have additional filters available. So if I open up stage, I can see that I have embryonic day 16.5 as well as 18.5. So if I was interested in just one of these 2 developmental time points, I could then apply that filter and see that now I have 18 data sets of this type. Sorry, not data sets, biosamples that fit that criteria. And now let me just drill down. If I want to know what information is available for this particular biosample, I have a link here, view, as well as under the RID, and this is just the record ID for this. And I can drill down here. I'm just going to mention that under this other column, we also have links here, but this will take us to the experiment. So right now, I just want to stay on this particular biosample. So let me follow that link down. So now I'm looking specifically at that biosample. What I see are additional details. I see it's a cryostat slice sample, and I see genotype and the litter number. I just see additional, whatever information was logged in FaceBase, I now can see the full details. I can also see the experiment that used this biosample. So now I'm going to go ahead and follow the link to the experiment. And now here I see that an RNA Seq assay was performed. I see a few additional details, of course, on it. And then I also, for this experiment, noticed that there were 4 biological replicates. So these are all going to be equivalent biological material used in this RNA Seq experiment. And then I can see the sequencing data, which these would be your raw reads. FASTQ files are generally the format that we take this in. So I can see I've got reads 1 and 2 here. I have processed data. You can see that I have gene counts and alignment tracks and other things like that. And then I get down to the actual track data, which you're probably going to experience that mostly through the Genome Browser, but it is available if you wanted to pull it down and create like a custom track or something like that for your own usage. Now, I would mention that both on the main dataset page, as well as here, because we have 4 biological replicates, I might not want to have to go through each of these files by file name to figure out what is, what 2 files. If I'm going to rerun this data, if you're going to rerun this data, rerun the pipeline for it, redo the process data and the tracks, you're going to need to know a few parameters, a few parameters for your pipeline. Those can be found up here in the summary of the experiment. And then you're going to need to know exactly which 2 files go together. And rather than making you figure that out based on file name, you can actually drill down under the replicate section. So let me go down one more level here under the replicate. And now what I see is specifically the two raw sequencing reads that are related to this one replicate. So I really know exactly what two files to put together in my pipeline. And then I can see specifically what process data, what tracks came out of those two original input files. So I get a better sense of where the data's at, how I could get it organized in a way that I could reuse it. And if I want to know more about the details of the process data on the tracks, I can see specifically what was the origin of that derived data. Now, again, these files are large, 2.5 gigabytes, 2.3 gigabytes. So again, you're probably not going to download from here. I will just point out that these are downloadable links. So in some cases with, say, a smaller data size, you might just grab them directly from here. So if I follow this link, I'm going to get a download dialog. Hopefully you see that. I don't know if Zoom captures that, but that's what I'm looking at. Okay, no, Chris says you're not seeing that. Well, it was a great looking file download window provided by Firefox. So that's what you get from all of these, but that's, again, probably not, especially with a large data set, the way you would want to experience this. Okay, so let me just work my way backwards here. And let me get back to that data set. Okay, so moving down this page, and on the left-hand side, I'm just going to use this table of contents to quickly jump down. So we just saw biosamples. Let me jump down to sequencing data. So here is all of the data that is in this data set. Now, again, this is the first 25. We can increase the number. I believe this one has, yeah, so it has within 100, so it's 76 records, and we could drill down into the Explore. But most likely, you're probably going to want to get this, if you're interested in drilling down, you're going to want to do that probably through the experiments or the replica and the biosample as a way to find more specific information of interest. So we have all of that here. And at least we kind of put this onto this page because, or at the top level of data set, because we wanted to make sure that everyone was aware when they came to a data set whether it had data. Because when you had to drill down only, there was room for confusion that maybe it didn't have data. So we want to make sure that's really clear. Okay, so back on the Genome Browser. So we have a link here to click here to visualize in a new window. This basically just pops out this display into a separate window. What I want to do is just jump over to a different tab I have open here. So I want to just look at another entry point really for FaceBase is coming through the Genome Browser. And you may have noticed when Chris was showing one of the earlier slides that we have a large number of data access for our genomic data. So we operate what's called a track hub, and that is a registered hub of genome track files. All of the data that gets submitted to FaceBase is if it's submitted with track data, it gets pushed onto our track hub, published out to our track hub, and it is then available for anyone coming to the UCSC Genome Browser or other, other genome browsers if you want to link to our track hub. But we're registered with the UCSC Genome Browser. So, for those of you who use this, you know probably where I'm going here. But under My Data, we have things like, if you record or take a snapshot of your sessions, you configure things just the way you want to see it and share that. I won't go into that. Also, custom tracks, or maybe you have some track data that you generated that you want to just visualize on the Genome Browser. And then in the middle here are track hubs, and that's where you find FaceBase. And so you begin here with a listing of all of the track hubs that are registered. And there's a search box here, so I'll just put in FaceBase. And we see we have the FaceBase hub, and there are going to be 4 different assemblies available. And I'll just click on Connect. And then this takes me to a page here that has an MM10 link. I'm sorry I clicked on that kind of quickly, but it has a short timeout. So if I don't click on it, it'll take me right to the Genome Browser homepage. I didn't want to lose my place. So now here we are on the, we've just drilled down into FaceBase data. Now this data set is showing up, or super track in Genome Browser terms, is showing up because I was looking at it before, and the Genome Browser records, your current state in a cookie. So it just had this open because of that, but generally it's not going to already have that open. But then I can go into the listing of MM10 super tracks, and I can turn them on if I like, and then click refresh to see them. There's a lot of files here, so I don't want to go through the load time of that. But let's just say we found a track of interest. So we're looking at, and I know I'm not in a really targeted location to show this, but let's just say you find a track of interest. You can click on the track, and this drills down into some display kind of information about what we were looking at. But the link at the top says FaceBase details. So this is, again, as somebody who, let's just say we came in directly through the Genome Browser, and now we found something of interest and we wanted to learn more about it. So we have a link here for FaceBase details. We click on that, and now we're back on the FaceBase site, and we're looking at just that track. And because things are linked together in that overall data structure, that data model that we looked at first in my session here, we see that our track data has a link back to our data set or to the replicate. So I can just click on the data set, and then here we are back at this overall data set, and we can now take a look through it and find out more of the FaceBase data, the scientific metadata that was behind that track. So for anyone coming through the Genome Browser, and again, we have a large amount of access, some 77,000 track reads in a year, so that's a fair amount. And so this provides a fair amount of traffic here. So having seen that, I just want to look at a second example, and I won't go in as great of detail. Okay, before I do, sorry, I just want to look here. I already showed the export menu, and then the other is the share and cite. So again, here is this in the live page here that we have the shareable links, and I'll just mention these at the top. So these are direct links within FaceBase. These are kind of the equivalent of an actionable accession number, if you will. And the top one says version. So actually our database takes a snapshot in time of every single data change to any record in our database. And so if you wanted to see something at a particular point in time always, you can do that through this link. And then an up-to-date, just kind of always up-to-date link is one with that's a little shorter. It doesn't have the extra identifier bits on the end. And then, as I mentioned before, for every data set, we encourage the citation by providing the formatted version of that per the standards on the Nature Publishing Group, and then the downloadable BibTeX file. Okay, so with that, let me just switch over to an imaging example, and then we'll move on to Alejandro's. So, whoops, that's not the imaging. Okay, here's the imaging. Okay, so we have another example here. So we again see a familiar organization with the overall title and identifiers, its own unique digital object identifier, a description, the attribution information, the project, the contributors to it. In this case, we also have a link out to PubMed for the publication related to this. And we also have a related data set. So I won't drill down, but this one has a companion data set that is RNA Seq data. And so from there, we can find out the underlying genomic information related in this overall investigation that was carried out. And then we have the experiment types. Again, if we click on these table mode versus custom mode displays, we can see the specific terminology used to describe this, and so on. Then we have a thumbnail. So a lot of data sets will have a thumbnail section, especially the imaging data sets. And then we next have, because this is, let me just scroll back up and point this out. So this is fluorescent microscopy, it's micro CT. There's also H&E data in this data set. So we have a variety of different data types. And so here we have a preview of the micro CT data here. And this is with our embedded micro CT viewer here. So we can rotate this around. You can zoom in and out on this, and we can also move through the slices of each angle of the image here. So we can kind of get a better sense of the data even before making the decision about whether we want to download and make offline use of this data. And then something that is sometimes missed here on the side is that we do have a little panel here, a little control panel here that allows us to switch into a 3D mode. So this is an on-the-fly, in the browser transformation of this image stack, because this is just like a TIFF, in this case, nifty image stack, that is being converted in real time. So it does take a little bit of time. So it's going through this processing step. And so now here we have this 3D rendering of this image stack. And it will do that kind of processing a couple more times, maybe one more time here. And then it'll stop. At that point, it'll stop doing that processing. It just does that when you change the, rotating around a particular axis. Okay, so that's the imaging data. And then of course, as we go down, we have the experiment and biosample and the details of the image files here. So that concludes my part of this talk. And I hope that gives you an idea of the overall organization and capabilities that are available in FaceBase for viewing the both imaging and seq data, as well as other types of data. I would just mention that for human subjects data, we can have a lot of that initial summary and even the Genome Browser and the scientific metadata, so you have a good idea of it. And then we just won't have the file data. And for that, you need to go through a data access request process. So with that, thank you for your time. And I'll turn it over to Alejandro.